30 years ago, I was training to be a neurosurgeon, and I met a patient named Janice who changed my life. I was prepared and accustomed to fractured skulls, gunshot wounds, brain injury, but none of that prepared me for what happened to Janice. Janice was 11 months old and was crawling across the kitchen floor as her grandmother was cooking dinner. She crawled under her grandmother's legs just as the woman turned to drain a pot of boiling pasta water into the sink. She stumbled, spilled the water on her granddaughter, and Janice's entire body was scalded. She was my patient in the burn unit for a month, where every day we changed her dressings, gave her antibiotics, did multiple skin graft surgeries, and hoped for the best, that she wouldn't get sepsis and an infection. And she did very well for a month, in fact, at the end of which she actually celebrated her first birthday. And the next day, I remember standing in the doorway to her room at lunchtime, watching her feed from a baby bottle, and thinking it was almost a miracle that we were even thinking about sending her home. Just then, her eyes rolled back in her head, and she went into shock. I ran into the room, cradled her in my left arm, and started giving her mouth-to-mouth, while the nurse called the cardiac arrest. The code went on and on and on. It seemed like an eternity. But it was no use. She was gone. She had died in my arms. It was the saddest thing. And worse, I had to talk to her family, and I couldn't explain what happened. She'd been doing great. There was no bacteria in her bloodstream. There was no signs of infection. There was no reason to think this would happen. And the more I looked into the problem, the more I realized that basic science lacked a fundamental understanding of why septic shock occurs. So 30 years ago, I decided to go in the lab and study that problem. And my laboratories have been pursuing that question ever since. We started with a large team of people at Weill Cornell Medical Center and at Rockefeller University. And what we found changed the way the world thinks about how the body responds to infection. We found that it was not bacteria directly that caused shock, but rather the immune system. The immune system producing a molecule called tumor necrosis factor, or TNF. Now, TNF is made by white blood cells and macrophages, and in small amounts, it's good for you. It's very beneficial. But if TNF becomes overproduced, it damages organs, causes blood pressure to plummet, and can cause lethal shock. Knowing this, my colleagues and I were able to develop monoclonal anti-TNF antibodies. And we gave these for the first time to baboons in septic shock. These were big experiments, and they would often go to the wee hours of the morning. And I can remember the first time we did this, it was about midnight, and there were 5 of us standing around, watching, astounded, as a baboon recovered from septic shock because we had given it monoclonal anti-TNF. Within a relatively short period of time, less than 10 years, monoclonal anti-TNF was approved for clinical use by the FDA, and now millions of patients with rheumatoid arthritis have received benefit from monoclonal anti-TNF. As gratified as I was by that, and as exciting as it was to be part of that, I found myself still thinking about Janice and what had happened. Why is TNF overproduced in some cases and not others? So in my lab, we studied TNF and tried to understand how the body controls TNF being made. And down the hall in my neurosurgery clinic, I examined patients with diseases of the brain and spinal cord. And I tested their reflexes. I tapped their knees with a rubber hammer. I stroked their corneas with a cotton gauze. And I knew that reflexes are absolutely critical to maintaining healthy organ function. And I knew that disaster strikes when reflexes fail. When the brain swells and bends the brainstem and cuts off those brainstem reflexes, patients can die from uncontrollable hypertension or apnea or cardiac arrest. Now, neither myself nor anyone else connected reflexes to TNF at that time until an accident happened in my lab. In the lab, we were studying rats with a stroke, and we were putting an experimental drug into the brains of those animals. And when we did that, as expected, putting the drug in the brain of the animals blocked the TNF in the brain. What we didn't expect was that the TNF was also blocked in the heart and in the gut and in the liver and in the spleen. This was baffling. It made no sense because there were no known connections from the brain to control the TNF production in the immune system. So my colleague and I searched for the answer to these connections, and we searched for the neural connections that could explain how the brain could control the TNF response in the body. And the search ended on a nerve called the vagus nerve. The vagus nerve is one of the most important nerves of the body. It's like a transatlantic undersea cable that carries tens of thousands of individual wires to all the organs of the body. It wanders through the body and brings these nerves to the organs. And at the nerve endings, those nerves control the activity of cells in the organs that keep them functioning in a healthy range. What we realized from a long series of experiments was that the vagus nerve was carrying the off switch. The vagus nerve was controlling TNF. And we proved this in rats by electrically stimulating the vagus nerve and finding that it turned off TNF in the body. We published that paper in Nature because we'd realized we'd discovered a reflex that controlled the immune system. We were incredibly excited, and I remember soon after at lunch, I wrote on the back of a napkin the idea to do this in patients. And we would put a small stimulator on the vagus nerve in the neck of patients and turn it on. And the electrons in the stimulator would send signals down the vagus nerve that would turn TNF off in the body. And I imagined that this might be a way for patients to have a device using electrons to replace monoclonal anti-TNF. So as excited as I was about this, in order to do a clinical trial, I needed much more. I needed to understand mechanisms. Mechanisms are key to understanding how to develop drugs or devices. And unless you can understand mechanisms, you can't solve the problems. So in the lab, my colleagues and I spent many, many years mapping in exquisite molecular detail the mechanisms to explain how signals traveling in the vagus nerve would control TNF. And what we found is that the signals were relayed to the spleen. And inside the spleen, those signals are converted from electrical to chemical. The chemical signals activated a white blood cell called a T cell, which also responded by making another chemical called acetylcholine. The acetylcholine targeted the macrophages at the end of the line, the macrophages making the TNF. The acetylcholine turned them off. So what we showed was that we could trace the electrical signals from the vagus nerve in the neck all the way into the cells making the TNF, and we learned how to turn it off. We were incredibly excited now to move this into the clinical tests. And what we did was start a drug company called Setpoint, and Setpoint set about doing the clinical tests. Setpoint recruited one of the leading rheumatologists in the world, Paul Peter Tak, to do a clinical trial of vagus nerve stimulation in patients with rheumatoid arthritis. Patients came into the trial with very high levels of TNF. They also had severe disability, severe pain in their joints, swelling, inability to move. Neurosurgeons in the trial implanted vagus nerve stimulators, much like I showed you on that cartoon a few minutes ago, and turned them on. And within days, TNF levels started to fall. And by day 42 in the trial, TNF levels were nearly normal. And more exciting yet, the patients felt better. Their pain went away. The swelling in their joints improved, and their mobility came back. This has been much talked about in the press, and one patient in particular talks about how some mornings before the trial, she couldn't grasp a pencil. She couldn't dress herself without assistance. She couldn't take long walks, something that she'd really enjoyed. But after the trial, she's off her medications. Her pain is gone. She's taking long walks. And she's riding her bicycle 20-mile round trips to the Dutch coast on weekends. It's fantastic. Now I get emails almost every day from patients who tell me they don't want to take their drugs. They don't want the side effects. They would rather have a device delivering electrons to replace drugs. It's a big idea. There are billions of neurons in the body that interact with almost every cell in the body, and at each of those nerve endings, molecular signals control molecular mechanisms that can be defined and mapped and potentially put under control. Many of these mechanisms are also involved in important diseases like cancer, Alzheimer's disease, diabetes, hypertension, and shock. It's very plausible that finding neural signals to control those mechanisms will hold promise for devices replacing some of today's medications for those diseases and creating new treatment options. This is an idea that has gone beyond the patient stories and beyond the basic research. Hundreds of millions of dollars of new research investment are pouring into this space from the NIH, from DARPA, from GSK, and from other companies. And more is coming from government, from academia, from private philanthropy, and from other industries. It's an incredibly exciting time for this field of bioelectronic medicine. And let me tell you what we're doing about it. We're building a new center, a major new center for bioelectronic medicine at the Feinstein Institute in New York, where we will, like the drug companies have done for 100 years, begin with the molecular mechanisms, begin with molecular medicine in a time that molecular biology is teaching us tremendous new insights into mechanisms of disease. We'll capitalize on those insights, but rather than screen for molecules to hit those targets, we will screen for neural pathways and neurotransmitters to control those targets, and then build devices to modulate those nerves to therapeutic advantage for the patients. We're well down the path to this process, and we're extremely optimistic. The story about TNF and the vagus nerve stimulator shows that this is feasible, and we're only limited by our ability to do research and identify additional new neural circuits and additional new targets. So I told you that I'm a neurosurgeon studying inflammation. Some of you in the room know that that's pretty unusual. And I told you why I'm studying TNF and inflammation. But something else happened that I didn't tell you, which is why I became a neurosurgeon in the first place. When I was 5 years old, we were living in Western Massachusetts in a small town with my father and my brother and baby sister. And my mother started getting headaches. My father drove us all down to Yale, where my mother's father, my grandfather, was a professor of pediatrics. Within a few days, my mother's life ended of a brain tumor at Yale. And I remember after the funeral talking to my father and my grandfather and saying, what had happened and why? And he began to teach me about the brain. And he taught me about its complexities. And he taught me about its importance in making us who and what we are. So for as long as I can remember, I wanted to be someone who understood the brain and invented things to help other kids with their moms so they wouldn't have to go through what I did. I don't think you can prepare a lifelong journey. I certainly didn't plan mine. But I do think that the combination for me of neuroscience and molecular medicine has put me in a unique position to think about, to imagine using technology to replace drugs. This is happening now. This is not in some distant, far-off time and place. It's happening today, and I'm very optimistic that some of the diseases my grandfather used to study, like whooping cough and polio, which you can only find in the back rooms of libraries and forgotten books now, that's what's going to happen to some of the diseases of our future society. I believe strongly that there'll be a time when Janice and many others like her won't die because of breakthroughs in bioelectronic medicine. Thank you.